Hi, Sally. How's it going? Can you guys hear me?
Yeah.
Okay, cool. Are you guys disappointed at the Chiefs loss? Or do you care?
Don't care.
Kelsey. What's that, Kelsey?
I care. It was pretty bad. Pretty bad game.
Was it? Yeah, I'm not a big football fan. It's interesting though on social media, like after they won the Super Bowl, everybody was like, "We won, we won," and I was like, "But you guys didn't do a single tackle or catch a single pass or anything else. You guys are just sitting at home watching on the TV." And then this morning and last night, this morning and last night, I look on social media and nobody's like, "We lost. We really let our team down." It's like, it was like you didn't do anything to win and you didn't do anything to lose. Yet when they won, everybody was like, "We did it." And when they lost, they were like taking no responsibility at all for the loss. It just seems weird to me. Anyway, my opinion about national sports. It's cold. It is cold. Questions from last time? Rose, are you back with us on campus?
No, not yet. It
No, not yet.
the snow didn't let me come back.
Yeah. Okay.
So
No, that brings up a point. So Kiara, you got my email, right?
Yeah.
Yeah. So we were supposed to go and do road cruising yesterday, and Kiara emailed me and said, "Hey, I can go tomorrow." She emailed me on Saturday, said, "Hey, I can go tomorrow," and then I emailed her back and said, "There's no point," for two reasons. But the first point that I gave her was totally valid, which is that they would have plowed the road, hopefully, such that any roadkill that had accumulated over the last 2 weeks was going to be scraped to the shoulder, so that one, it would be really hard to find because it's probably covered by snow on the shoulder, and so we wouldn't get an accurate count of what roadkill was there. And the second thing is, it's also important, one of the things that we collect data on is where on the road it is at. Is it in the driving lanes, such that people drove over it in the driving lanes, or was it actually killed on the shoulder, which means that somebody actually swerved out of their way to kill it, which happens a surprising amount of time, as it turns out, when you start actually gathering data on it. And so since all of the roadkill would have been on the shoulder, we wouldn't have an accurate view of whether things were being killed in the driving lanes or killed on the shoulder. And then I went to bed and woke up the next day and saw that it had snowed a lot during the night. And so then everything was just covered in snow. So we wouldn't have been able to see roadkill anyway, even if the roads hadn't been plowed. So look at the schedule. A couple of people have already responded. The next time we have scheduled, generally when I miss a day because of weather, I just basically give it 2 weeks after that. Because if they're plowing the roads now and they continue plowing the roads this week, there won't be the accumulation because those things keep getting knocked off of the road. So typically, I just stick to the schedule, and then we just have this month-long gap in the data. And that's one of the problems with field work, is that you occasionally get gaps in your data that you don't have any control over. So that's just the status of that. Okay. Questions from last time. I realize it feels like it was forever ago now. What screen are you seeing?
Like your home screen with the file window and the meeting recording.
Oh, with the file window. Okay. All right. That was not what I intended. All right, now which one are you seeing?
There's a woodpecker on it.
Okay, and now it has a car on it, doesn't it?
Yep.
Okay, good. Okay, that was part of something I was doing for my stats class. So let's reduce that. All right, so where were we last time? Who did we end up with?
I think we ended on the Hylidae.
Well, we hadn't gotten to the Hylidae yet, had we?
Yeah, I think I went through what I had.
Oh
to
yeah,
talk
because
about.
we did talk about the disc, the toe discs. Okay, so we were about to move on to the Bufonidae. Who had the Bufonidae?
I did.
Okay, talk to us about Bufonids.
So the Bufonidae are the true toads and harlequin frogs. They vary from 20 millimeters to 23 centimeters in length. They have a bitters organ, which is an ovary that develops on the anterior end of the larval testes, and it's a paedomorphic trait. They don't have any teeth. They have a heavily ossified skull, and the skull is sometimes co-ossified with the skin. And then they have prominent parotoid glands. And there's 47 genera and at least 590 species. And they're pretty much found everywhere except for, if you look on the map, where it's like too cold or too hot, like in the Sahara and then at the poles.
Yeah. Mm-hmm. Yeah, so strangely enough, they're absent from Europe, which is unusual. But yeah, they tend to be I don't want to say kind of tropical, subtropical, but they don't really like super cold. Well, sorry, that's Microhylidae. Bufonidae. So I was looking at the wrong group. So they do get up into northern areas. They're found in the Rocky Mountains. We've talked about the Colorado toad, which is a Rocky Mountain toad. So they're found throughout a wide range of areas. They're not found in Australia, largely because of biogeographic reasons, and they're not found in really harsh deserts. So pretty wide-ranging group. As you look at toads in the lab, what are the kinds of characteristics that you're having to pay attention to when you look at the toads?
Parotoid glands.
Okay, so the position and size and placement and shape of the parotoid glands. What else is very useful for looking at toads?
Does that have something to do with their the size of the warts? Like with the Americanus, the warts on the back legs are normally larger than the ones that they have on their body.
Yeah, so the wartiness, the presence of these granular glands as they're distributed around the body is also important. But also, what else is up near the parotoid glands that is useful taxonomically?
Cranial crust.
Yeah, these cranial crusts. So if you look at Ancilius valliceps, one of the things that you'll notice about Ancilius valliceps, this is one of the toads, it doesn't occur here, but it occurs further south. You see that the degree to which the cranial crusts are prominent and also the shape of them. You see how in this one, the cranial crests come around on the front side of the eye and the back of the eye. You have spurs that go off the backside, and there's this deep valley between the cranial crests. That actually is a species characteristic of this particular this particular species, the Gulf Coast toad of the United States. So this ossification that, Elizabeth, that was you talking, right? Yeah, this ossification that she referred to is largely referring to the presence of these cranial crests and the shape of those cranial crests, what their relationship is spatially to the parotoid glands, the size and shape of the parotoid glands. All of these things have taxonomic importance and are things that you should pay attention to. If we had a wider diversity of toads in Missouri, the importance of the cranial crests and the parotoid glands would be more obvious than it is right now. But the fact of the matter is the toads that we have in Missouri were all members of a single species that has now been split up into what were formerly subspecies. And as a result, they're actually really similar, which is why toads tend to give students the most problems when we're covering the amphibian groups. Where do these guys breed? What's their breeding like? Anybody? Elizabeth, at the very least, should know.
I didn't see it in the reading.
Okay, so these are going to be mainly pond breeding species. Very rarely do they breed anywhere other than still water in things like ephemeral pools. Remember that when I refer to, have I used the term ephemeral pool before? No. An ephemeral pool is a temporary water source. One of the reasons that amphibians tend to breed in ephemeral pools is that they don't have large predators like fish because they're ephemeral. They dry up during part of the year and then they fill back up when seasonal rains come. And as a result, there are no fish in there, so it reduces the number of predators that are around to feed on eggs and developing larvae. And so many of these species will breed in ephemeral pools. The Plethodon and salamanders, for example, tend to breed in headwater streams that are so far up mountains and whatnot that there aren't any fish in those pools because they're just not deep enough to support fish. And they also breed in these seeps, like I talked about when I talked about going collecting salamanders in the Appalachians. And so those are, once again, relatively predator-free because they lack fish. And so bufonids are not that different from many of the other frog groups in terms of where they go about breeding. All right, who had the microhylidae?
I did.
Rose, talk to us about microhylids.
So they're also known as the narrow-mouthed frogs, and they are geographically widespread, and there are 683 species and 11 subfamilies, which is the largest number of genre of any frog family. And they have a variety of reproductive strategies, including aquatic larvae without mouth parts, and direct development. And they also live in a variety of habitats, from deserts to wet tropical rainforests.
Here in the United States, we have a couple of examples of narrow-mouthed frogs. We have Gastrophrine olivacea and I'm forgetting what the species name is of the other one. But the Gastron olivacea has actually been found commonly down in the burrows of tarantulas. And when you dig down into a tarantula burrow, it's basically just a silken tube that has been dug down under the ground, and then it opens out at the bottom into a slightly larger living space. And oftentimes, if you dig up a tarantula, you will find a narrow-mouthed toad sitting at the bottom of that tube, living as a commensal with the tarantula. The tarantula doesn't tend to eat it, and it tends to the frog will tend to eat any pest insects that end up falling into the tube that the tarantula lives in. So it's just an interesting commensal relationship that you don't find very commonly. What do these guys eat typically? Rose, did you find anything about that?
No.
Yeah, so these guys have very small mouths, and the mouths are at the very tip of their snout. They don't have large mouths. They don't have a very large gape. And these guys are oftentimes specialists on things like ants and termites. And so in terms of dietary specialization, this is one group that as a family tends towards dietary specialization, unlike a lot of other frog groups. So for example, a bullfrog will eat anything that it can fit in its mouth in the family Ranidae. Same thing with toads. They tend to eat insects, but that's generally because insects are of the size that they can fit in their mouth. But for example, bullfrogs, you can feed bullfrogs small rodents and things like that. When they get big enough, and they'll handle them just fine. So dietary specialization isn't super common in frogs. They tend to be generalists, but there are some groups that are more specialized, and this is one of them. All right, who had the Ranids?
I did.
All right, take it away.
So these are the true frogs, and they have the widest distribution of any frog family. And they're on all continents except for Antarctica. So if you look at the map, although they're only like a little bit on Australia, they have like a pretty wide range of like habitats and ecoregions and elevations, pretty much covering all of North America and all of Europe, most of Asia. And they have smooth and moist skin with like large and powerful legs. And they also have extensively webbed feet. And they vary in size. They're the generalized pond frogs. And then it's the lithobates in the field guide and in the like lab we were looking at.
Yeah, so all of the North American frogs in this family were in the genus Rana. And your textbook, which has not been updated in the time since your field guide has been updated, their taxonomy has changed. So Lithobates is the current genus for the North American representatives of this family, and they're no longer Rana. So when you see, for example, the example Rana catesbianus in the textbook, just make that translation to Lithobates in your head. The old world species are still in the genus Rana, but once again, taxonomic revision. Where are these guys going to breed, Kelsey?
I don't know, in a pond.
Yeah, they're pond frogs. So we're actually going to go out once the weather turns nicer. We're going to go out to Cooley Lake. Do you guys know where Cooley Lake is? Yeah, it's on the way to Orrick. You pass Missouri City, and on the left, there's a big hill to the north side of Highway 210. And there's a spot there that they used to use for as kind of a fish hatchery in an old oxbow lake. And the old oxbow lake is called Cooley Lake. And there's a big bluff above it, and there are a lot of snakes there. And down in Cooley Lake, there are gajillions of bullfrogs that you will find in the spring. And so we're going to go out to Cooley Lake and do herping one day. Herping, Kelsey, one day during lab.
Not a verb. I don't like it.
It is a verb. Talk to any herpetologist and ask them if they've been herping lately, and they will be able to tell you whether they have or not. So we'll go collecting amphibians and reptiles there, and we will almost certainly see Lithobates catesbiana there. And they have large masses of eggs. The way in which frogs and toads lay eggs can be taxonomically informative. So in the case of the pond frogs, they lay these big masses of eggs where the eggs are all stuck together. But in some cases, frogs will lay long strings of eggs, such that it's like a necklace of eggs in a long string with each individual egg following the next. They just vary quite a lot in terms of their egg laying morphology, if you want to think of it that way. So that's the frogs. And once again, we didn't cover all the frog families, but we covered the ones that are in North America and ones that are of note from other places. We didn't cover things like the dendrobatids. So there are frogs in the tropics that, this is the family Dendrobatidae. And I can't believe that they didn't make my list. These are the poison arrow frogs. They're found on page 78 of your textbook, New World Tropics. Once again, the main reason that I'm calling attention to them is because they have interesting predator-prey interactions. They tend to be brightly colored. They're ant specialists in most cases. And the toxins that they produce are derived from the compounds that they ingest when they eat ants. So as soon as you take them away from an ant-based diet, they tend to lose their toxicity because they're using metabolites gained from eating ants that, in terms of creating their own toxins. So I think that's the only family that I wanted to call attention to at this point that we didn't already cover. All right. Questions about frogs and toads? All right. Let's briefly talk about caecilians. Hopefully you looked in your book at caecilians and noticed that these are long limbless creatures. They're burrowing. They can be either fully aquatic or they can be terrestrial. They are among the most primitive of the amphibians. So if you look at the phylogeny, how far back? No, I don't have that phylogeny. Well, you'll see that they're a stem group. Now I have to get back. So oftentimes they don't have they don't have pectoral or pelvic girdles. So they've lost not only the external portions of their pectoral and pelvic girdles, but they've also lost the internal portions. One of the things that you'll find with snakes when we get to snakes is that the snakes have, in some cases, vestiges of the the pelvic girdle in some groups of snakes, the boas and pythons. in many cases, probably just an adaptation to a burrowing lifestyle. They will have reduced eyes and reduced visual acuity also. And then in a kind of throwback to their evolutionary precursors, in some groups, they do have dermal scales. So when we were going through the dichotomous key on Thursday, I started to add something to separating amphibians from reptiles. We had amphibians were scaleless and lacked claws. And I almost wrote out there, if they have scales, the scales are dermal rather than epidermal in origin. And then I erased that because that applies to caecilians, and we don't have any caecilians in Missouri, and we're writing this key, this dichotomous key for the amphibians, reptiles of Missouri. So when you look at reptiles, which we'll look at after we finish with the caecilians, reptiles have scales, but those scales are epidermal scales. They're derived from the upper layers of the skin. Some caecilian groups have scales, but they're derived from the dermal layers of the skin, the deeper layers of the skin. And the scales that you see in fishes are dermal scales, not epidermal scales. So the scales that you see in caecilians are much more similar and homologous to the dermal scales of fishes than they are similar to the epidermal scales of reptiles. So if you ever find a caecilian and it has scales, don't get confused and think that it's a lizard. They only have scales also in these things called the annular grooves, which we'll look at when we see some photos of them. There's a chemosensory organ up on the head, referred to as a tentacle. And one of the characteristics of this group is that the skull bones are largely fused, and they basically totally enclose the head of the organism. And once again, this is probably an adaptation to a burrowing kind of lifestyle. So these guys reproduce with internal fertilization. So they have a copulation, they have a copulatory organ called the phalloidium, and it protrudes out of the male. So it's an erectile organ, and basically copulate with female the way any other organism with a penis-like structure would do so. The caecilians are interesting in that they have interesting reproduction in terms of they tend to retain their eggs. In some species, they tend to retain their eggs in the oviduct. And what happens is the eggs will actually hatch within the oviduct. The offspring will then feed off of secretions that are secreted by the oviduct, and that will then feed the developing larva. And then the larva, when they're big enough, will be born through live birth. And so that's a common way in which the caecilian larvae get their nutrients. Rather than their provisioning of a large yolk, they'll actually produce more eggs with smaller yolk, and then the female will keep them inside of her and feed them off of the lining of the oviduct. In some groups, the larvae will hatch out, and some of the eggs will be consumed by the developing larvae. And in at least some cases, the female actually produces unfertilized eggs for this purpose. So they have a very different way of going about reproduction. So this is kind of what they look like. And once again, you can see they have really small eyes. You can see the tentacle here. You can see a tentacle here. You can see a tentacle here. In all cases, the eyes are reduced. You can see that this eye is covered with skin, so it doesn't really function any function other than just light sensing. It's not really forming a focusable sort of image. And you also will see that the eye, in some cases, can be associated with the tentacle. And so in some species, the eye is quite far from the tentacle. In some species, it's closer. And in this particular species, the eye is actually right down at the base of the tentacle. And so once again, that becomes important taxonomically. In this upper picture here, you can see these annular folds on the body. They start just behind the head. These are the annuli. So when we talk about where these dermal scales are present, they're in the grooves between the annuli. And once again, if you see a long snake-like organism that has permeable skin, not scaly skin, and is divided up into these annuli, that is He Caecilian. This is looking at the skull, and as you can see, the skull is heavily ossified. And once again, this is probably an adaptation to burrowing. You'll see that in many cases, the nose, the head is rather flattened. And once again, that enables the animal to basically push its nose into soil, flatten the soil to the dorsal side and the ventral side of the animal, and then the animal can push further into the soil. So these are definitely adapted to burrowing. And if you had a more kinetic skull, a more loosely articulated skull, that would be more difficult. But this skull is fused and basically becomes a burrowing organ. And we're not going to go into a lot of detail about Caecilians because, quite frankly, they're not really found in the United States or North America very commonly, and they certainly don't occur in Missouri. All right, questions about amphibians in general? All right. Recall that one of the ways in which I will test you, I talked about this in lab on Thursday, is I might give you matching a group of taxa on one side and a set of characteristics on the other side, and your task will be to match the correct morphological or behavioral or reproductive characteristics with the correct taxa is one way of testing this type of material. So our next task is reptiles. So just to get a brief introduction to the reptiles, this PowerPoint is up on the Moodle site. So if you want to go and grab that from there, you can. For Wednesday, I will do something similar to what I did with the frogs, and that I will assign you guys some families to briefly summarize, and we'll do a similar sort of thing like we did with the frogs. All right. Just here at the get-go, what would you think some of the differences are between amphibians and reptiles? At the very least, we talked about this in lab.
Scales.
Okay, scales. They have scaly skin. Once again, these scales are dermal or epidermal?
Epidermal.
They're epidermal, okay? What does that enable reptiles to do that amphibians cannot do?
Doesn't it have to do something with the temperature? They can, what's it called?
Not temperature as much as something else. If you have a hard, scaly skin that is relatively impermeable to water, what does that allow you to escape? Or you can think of it as not escaping something, but being able to take advantage of one type of habitat that an amphibian can't take advantage of as easily.
They're
Sorry.
mostly terrestrial. Oh, sorry.
Yeah, so these guys can be more terrestrial because they're not as closely tied to water because their skin isn't as permeable. What's another thing that separates reptiles from amphibians having to do with how tightly tied to water they are? Has something to do with the egg.
They have a shelled egg or like a parchment egg.
Yeah, so these guys have shelled eggs. They also have an egg that has an amnion in it, this extra layer, extra embryonic membrane that once again protects the embryo more. So one of the big differences between amphibians and reptiles is going to be that the transition from amphibian to reptile evolutionarily is also a transition from being tied to aquatic environments, both for reproduction and during your life, to being free from being tied to aquatic environments. And that is seen most evidently in the structure of the skin and in the structure of the egg and how they go about reproduction. So the skin is super important. The stratum corneum is this keratinized layer of skin, of epidermis, that is part of what makes these guys more impermeable to water loss. This is modified into scales, which are these folds of the epidermis. You find scales in all of the reptiles, even birds. So if you go and look at the feet of a bird, those scales that appear on the feet of birds, those are epidermal scales that are homologous to the scales of the other reptile groups. In addition to providing resistance to water loss, they're also resistant to abrasion, and beta keratin is found in these layers, the scales. There are some groups that have what we call osteoderms. So the crocodiles will have scales that are underlain by a bony component to them. And so you'll see these scales that look like flattened plates on the backs of crocodiles and alligators, and each of those large scales is underlain by a bony element. And then the turtle scutes are unique in terms of scales, in that their scales are coordinated with, once again, the underlying bone. And so scale structure does vary quite a lot across the different reptile groups. So this is some variation. This is a lizard scale. Actually, this looks like a bearded dragon in reality, from the looks of it. These are all epidermal scales. These are epidermal scales on one of the pond turtles. And in this case, the scales are not tightly tied to the bony structure that is underneath. So you'll find that this scale is actually overlying two different bony elements. But when you get into the tortoises, you'll find that each scale actually covers the area of an underlying bony element to it. And so turtles don't always have their scales coordinated with the underlying bony elements, but in some groups they do. And then this is an example of those osteoderms that occur on crocodilians. And so this large scale, if you were to dissect this specimen out after it died, you would see that if you cut through the scale and peeled this back, that there would be a large bony plate that is underlying this scale. Not so with these smaller scales in between. They would only be underlying these bony plates. These large scales would have a bony plate under them. So because you're stuck within this relatively inflexible scaly skin, you have to shed that skin periodically. And if you've ever seen a snake shed, in snakes, they just kind of pull their bodies out of their old skin like pulling a sock off. But turtles and crocodilians and birds shed their skin in clumps, and then the lizards and snakes generally lose all of their skin, all of their epidermal layers at one time period. In some groups, even though they lose it synchronously, it comes off in big sheets, whereas in the snakes, it comes off at a single time, generally in one single piece. A guy that I know just published a paper about the microstructure of snakes that do sidewinding as a method of locomotion, and all of their data on the microstructure of the snake skin was taken from shed skins of animals from zoos. So this implies a very different structure to the skin than what we saw in the amphibians. So down here at the bottom, we have the dermis, and then there's a base layer called the stratum germinativum. The stratum germinativum is just like what it says. It's basically just a layer of cells that are basically going through active cell division, which produce new cells of skin upward towards the outer layers of the skin. But this is produced in a couple of different layers. And so you find that there is a layer separating the alpha layer from the beta layer, and then there's another level of cells separating this outer layer from the beta layer. And what you have here is you have up here, this is the outer skin that is currently functioning as the skin. And then between these two alpha and beta layers here, there is a layer of cells that are basically going to be responsible for separating the outer skin from the inner skin. So the stratum germinativum is producing these two layers of skin underneath the skin that is about to be shed. This is the skin that currently is functioning in the role of skin. And you can see that the stratum corneum, the epidermal cells that down here have nuclei as this skin is developing, as the skin is moved out to form the outer layers, the stratum corneum, those cells lose their nuclei in the same way that your outer skin cells have lost their nuclei. And then on the surface of this, there is a coating on the very outside of this referred to as the Oberhauten or the cuticle. So we have the outer skin, the inner skin. The outer skin is currently functioning as the abrasive resistant type of skin. And when it comes time to shed, this skin will be lost and this skin will replace it, while then the stratum germinativum produces another layer of this internal portion of the epidermis. So substantially more complicated way of dealing with skin than we had to deal with in the amphibians. Scales, once again, are just folds of the epidermis, and they can be scales that occur as just bumps with little valleys between them, or you can have full-on projecting scales like you see, for example, in the fringe scales of horned lizards and in bearded dragons and things like this. Now, given that you have these thick layers of skin between the outside of the animal and, for example, the dermis, you have two layers of skin, one of which is going to be shed and then replaced by the lower levels as new skin is being produced. What are some challenges that this poses to an animal in terms of its ability to interact with its environment? Where are nerves going to be in all of this? Who's had developmental biology with Dr. Vora?
They're deeper.
Yeah, so where do we find nerves? Do we find nerves in epidermis or in dermis?
In dermis.
Yeah, we're going to find the nerves in dermis. So the nerves are way down here in the dermis, separated from the outside world where you might get sensory input by all of this epidermis. And so one of the ways in which reptiles have dealt with this is that you will find that there are sensory papillae that actually are sections of the skin, sections of the scales, where the epidermis is substantially thinned out, and they will oftentimes have sensory hairs associated with them. And at the base of this sensory hair will be the end of a dendrite of a sensory neuron. And so the epidermis of a reptile is not uniform in terms of its thickness. It can be thicker in places and thinner in other places. One of the places that it is thinned out is any place where there's a necessity for sensory input. You will also see that this outer layer, the oberhaut, can have various types of microstructures associated with it, and that's what's shown in panel C here. This is what microstructures on different kinds of snakes look like. When you look at it under a scanning electron microscope, this is a figure from a paper, I forget which paper it is, but once again, there can be taxonomically important differences in microstructure. This is once again the Oberhauten layer. This is the outer cuticle layer on the very, very outside, which is illustrated in figure C here. This is all the microstructure that you see, and it can vary quite a lot from group to group. Geckos, which I am interested in, are interesting because among the things that they have are the setae. The setae are long hairs, and when you look at the setae, you can see that this is a seta, and you can see that there are these microscopic hairs that come off of it. And these hairs are what enable geckos to stick to smooth surfaces. And it's all accomplished, it turns out, by van der Waals forces. So these weak molecular forces that you study about back in basic chemistry, the van der Waals forces, they're present on each of these little tiny microscopic hairs. And each of those little tiny microscopic hairs only has a little bit of force applied by these van der Waals forces, but when you add the cumulative effect of it over the millions and millions and millions of the hairs on these setae, you actually get a lot of ability of the gecko to cling. I have a Madagascar day gecko at my house, and it walks around on the glass constantly. It spends most of its life outside of its bamboo tube, adhering to the glass. But one of the consequences of this is that the placement of the toe is very important. So this is a scale. This is the Oberhauten extension of the lamella, and then you have the setae on the lamella. So the placement of the toe and how this toe interacts with the surface that it is on becomes very important. And so what you'll find on gecko toes is on the leading edge of the scale, there is a sensory hair attached to a sensilla underneath it that then has a nerve cell that basically communicates sensory information from that sensory hair. And you will find, so this is a scanning electron micrograph that I made back when I was in graduate school of a Mediterranean gecko toe. And each of these things is scale, and the end of the toe is down here, and the body is up here in the upper left. So the proximal end is upper left, the distal end of the toe is lower right. And you can see that at the distal end of every scale, there are a set of sensory hairs, and you can actually see that these hairs are curled down such that this hair and this hair and this hair makes contact with the scale that is next most distal to that scale. And when I was in graduate school, I hypothesized that these were actually sensory hairs that gave proprioception information to the gecko about what the position of the toes was, and very detailed information about where the specifics of the toes were, because it's so important to geckos to be able to position their toes with some precision in order to get this clinging ability. And then in 2009, some guys came along and basically showed that that is actually what is happening in these species with toe lamellae. They did it with anoles, but the same thing would inevitably apply to geckos. Once once again, geckos and anoles are in different families, and so these toe lamellae are adaptations that are convergently derived. They're not, anoles don't have toe lamellae because geckos are their ancestral group. These toe lamellae evolved independently in these two lineages. So these are examples of those thinning portions of the epidermis that allow for sensory information. And you can see that they look kind of like this. You have this papilla that sticks up, there's a valley around it, and then the normal scale continues in the same way that you have the normal scale, then you have this valley around it, then you have the papilla and the hair coming out. If you look at the distribution of these kinds of sensory organs on gecko toes, you find that in different parts of the toe, you have bristleless ones that are located mainly down the center of the toe. As you get out here to the edges, where you need more information about the position of the toe, those sensory papilla tend to be bristled. They have that sensory hair in the middle of them. And this is a paper by Aaron Bauer, and I forget when this was published. So basically, this is a schematic showing you what I was referring to before. Over time, you get cell production by the stratum germinativum to produce new skin. The skin differentiates into the stratum corneum and the nucleus containing cell layers beneath that. You then get this setup of a boundary layer between what is the outer skin and the inner skin. The outer generation, the OG and IG from the previous slide refers to the outer generation and the inner generation. And then there is this layer that separates those two, and it's that layer that is then going to provide the separation between the outer generation being shed and the inner generation that is retained after an animal goes through a shedding phase. And then at this point, you're back to this stage here, and then you go through the cycle again. So these are just examples of sheds. Once again, a snake tends to shed all in one big piece. In lizards, the shedding is synchronous, but it also often doesn't come off in one big sheet. It just comes off in these big patches. And depending on the group, in some groups it just sheds and falls off, and in other groups, the animal will actually eat its shed and recycle some of the resources that are in that shed. So these scales present a challenge to sensory perception. So some of the things that these organisms have that help them with this are these lenticular organs that we've talked about before, these sensory papilla. But then some groups of reptiles have really highly developed senses of smell. In the case of snakes, they have a Jacobson's organ, which we'll discuss in more detail when we get to the snakes. But in the snakes, you have a hard palate. So you don't have to do this on camera if you don't want to, but you can. If you put your finger in your mouth and you feel the roof of your mouth, you'll feel that there's a hard palate there. It's skeletal. But if you go far enough back, you'll get to the very back of your mouth, and there's a point where the roof of your mouth is no longer underlain by bone, but it's just underlain by connective tissue. And you can feel it back there, and it will become more pliant as you poke up towards your nasal cavity. And so you have a skeletal structure in your skull that has a hard palate up front and then a soft palate in the back. So in the front, where the hard palate is, it's underlain by bone, and the soft palate in the back is not underlain by bone, which is why it's soft. In snakes, they also have a hard palate, but the hard palate is way forward, and they don't have a soft palate because the soft palate has disappeared. And so at the back of their mouth, there's this direct communication between their oral cavity and their nasal cavity. And that cavity, where there is no soft palate, is the Jacobson's organ. And so in the mouth of the snake, they have direct communication with the olfactory mucosa. So their scent glands, their scent structures, the neural structures that detect olfactory cues, are in direct contact with the mouth. And when snakes flick their tongue, they're picking up scent particles on the tongue, they're pulling them into the mouth, and they're putting those two projections of the forked tongue up into contact with the nasal mucosa in the Jacobson's organ at the back of their mouth. And that then brings the particles that they're sensing into direct contact with the olfactory organs of the nasal mucosa, and it gives them an enhanced degree of scent. And so you will oftentimes see a snake project its tongue out and actually touch the substrate in front of it and then pull it back in. And when they pull it back in, they're taking whatever they picked up off of the substrate, and then they're waving it around and touching the nasal mucosa, transferring whatever they picked up from the substrate to the nasal mucosa for smell. When you see a pit viper strike a mammal, strike a prey item, what they do is they strike the mammal and then they pull back. They've envenomated the mammal, and then the mammal hops off and goes somewhere and dies. Well, one of the things that you'll see immediately after a rattlesnake strikes a kangaroo rat or something is their tongue flick rate goes way up. And they are tongue flicking because the animal has run away. It's been envenomated. The animal runs away, but it then starts to track the mammal by scent cues, and it's following two things. It's following, one, the taste of the animal that it just bit, but it's also probably tracking the scent of its own venom. And so I'm going to shift things here to a different view if I can. So can you see, I'm going to just stop sharing actually. Can you see my screen or my board pretty well? So one of the ways that you can test this trailing behavior in snakes is you can put them in a room or in a container. I did this in grad school in a container, a big fiberglass box. And you can put the snake here at the end in a container, and you can have a door that slides open and closed, and you can put a mouse in front of the door, and then the snake will strike the mouse, and then what you have drawn on the floor of the container is a path, a circuitous path. And then you can have a box here and a box here. And when the snake has struck the mouse, you then close the door so the snake can't see where the mouse is going. And you can drag the mouse along this path. And you can deposit the mouse in the closed container at the end. And what you will see when you then open the door and let the snake out, the snake will go along and its head will go back and forth, and it's tongue flicking the entire time. And as it gets away from the path of the mouse, it's not picking up the scent particles anymore, and then it goes back to the left. And then as it goes to the left, it gets out of the scent trail and then goes back to the right. And when it comes to a corner like this, it will oftentimes overshoot the trail somewhat, but then it recognizes that it's not picking up the scent any longer, and it will start searching back and forth. It'll then pick up the scent, and it'll turn, and it'll go down this way. And you can calculate the degree to which the animal is capable of scent trailing scent trailing prey item by looking at how much time its head is outside of the path versus how much time its head is inside of the path. And it turns out snakes are really effective at scent trailing prey items after they have envenomated them. We're almost out of time, so I'll just tell you a little story. I was biking across Missouri back in 2006. And when I got onto the Katy Trail, headed towards St. Louis, it was September. And that's the time when snakes are generally moving to their hibernacula, their underground dens where they're going to wait out the winter. And what typically happens with copperheads is right before they go into hibernaculums, they generally mate before they go into the hibernacula. They start incubating their offspring over winter, and then when they emerge in the spring, they're gravid, and then they give birth late spring, early summer. And during this time, during the fall, when they're going back to the hibernacula, the males are trying to mate with females, and you will oftentimes see males trailing females based on the scent of the female. And I saw a surprising number of copperheads on the Katy Trail, especially as I approached St. Louis, as I got closer and closer to St. Louis. And I was riding along one day and I saw a nice big female just kind of extended across the Katy Trail. So the Katy Trail is a bike path, and there was a nice long female. And I stopped and took a couple of pictures of her. And as I was taking pictures of her, she slithered off the trail to the south. And after she went off into the grass, I went back and got my bike. And as I was getting on my bike, another copperhead emerged out of the forest on the left-hand side of the trail. And it was a male, and it was basically following the exact path that the female had taken across the trail. So they'll use their scent, their olfactory abilities to not only track prey that they have struck, but they will also use them to track potential mates based on pheromone cues. And once again, their sense of smell is very good, and they're very effective at this. Questions? We will continue with the reptiles on Wednesday. We'll get as far as we can get through the reptiles on Wednesday, and then that'll be the cutoff for information for your first lecture exam, which is scheduled for Friday, correct? Yep, okay. So whatever we finish with on Wednesday, that'll be on the test, and whatever we don't get to, we'll just cover after the test is over on Monday. All right.
You said you were going to upload an exam, right? So we could get
Yeah, and I didn't do that yet. And as I was talking about it earlier, when we finished the amphibians, I thought to myself, oh yeah, I said I was going to do that and didn't do that. So I'll try and get that done today.
Okay, thank you.
Sure.
Paul, can you open the lab tomorrow morning?
Yeah, sure. What time do you need it open?
Like 9 or so is fine.
Sure. Yep, I can do that.
Okay,
All right.
thank you.
See you guys.